All right, everybody. Thank you for coming in person in Zoom land. It's my great honor to introduce a good friend of mine, Dr. John Couture. He is at Purdue University with a split appointment in entomology and forestry and natural resources. He got both his bachelor's and master's from Western Michigan University, and then we did our PhD at the same time in Wisconsin. So go Badgers. John has, from what I looked at, he does a lot of cool stuff. I kind of boiled down what he does into three main things. Hold on, nail it here. Chemical and landscape ecology, broad stuff. Environmental, the impacts of environmental change on plant and insect ecology, which is kind of what we did when we were there. And then what we're going to talk about today, he's going to talk about today, is using hyperspectral data in the grander scale of figuring out plant and ecosystem health. So with that, John.
Welcome. Thanks, man. Thank you very much for the invite, Dave. I'm very happy to be here. It's beautiful campus. I love the rolling hills. Very different from West Lafayette, which is as flat as this floor is. So I was thinking about around this morning. So today I am going to talk about how do we use hyperspectral data to understand plant and forest health. And I usually start with this slide because it helps me give context to a lot of the thinking that I do about this. And it's we're at a very interesting time in plant ecology, right? Never before have we had access to such level of omics style data. Think of chemistry, think of genetics, think of genomics, think of CRISPR, think of all of the advances that we have when we can manipulate plants and understand them. But really never before have we needed a lot of those approaches because of the grand challenges, sad grand challenges that we're facing, such as shifts in biodiversity, losses in biodiversity, climate change, fossil fuel emissions, and ultimately needing to balance how do we manage many of our natural and forested areas with the need for an increase in agricultural production to meet a growing population. So what we need to do is to develop questions that can occur at multiple spatial scales. So we can start to blend these different levels of details of information together. So we need approaches that we can use at the suborganismal level, such as the leaf, whole organismal level, so the canopy, and then we can actually scale up to landscape measurements as well. So we can start to ask questions about how do individuals respond to stress, how do populations respond to stress, and ultimately how do communities respond to stress. And the approach that one of the approaches that my program does is we use spectroscopy. Now, it's been used in geology forever. And at the brass tacks, it's just the way light interacts with the substrate, right? And you can pull information from that about in vegetation. And if you look, this profile is what a green reflectance profile looks like. This is from fresh living tissue. We usually focus on the solar spectrum, so about 350 to 3,000 nanometers, because that's the radiation that vegetation uses. And for vegetation spectroscopy, changes in these spectral profiles, and you'll see shifts in spectral profiles as we move through different examples. They're caused by variation in biochemistry, biophysical properties, structure. Interestingly, much of the information is contained outside of what we see. So think of your phone taking a picture. You typically have like a 3-channel imaging system in your phone. There usually is in most iPhones a 4-channel that if you disable the filter, you can use. But what we see is this blue, green, and red. And this peak in the green right here is where chlorophyll reflects light, absorbs in the blue and the red, which is why plants look green. Much of the information, though, and quite a bit of the important information is outside of that area, right? And this is the visible wavelength range, which is what we interact with our eyes. We also have the near infrared range, which is characteristic. There's some water absorption features, some protein absorption features. A lot of structure is contained in the NIR from a biological standpoint. And then you have the short-wave infrared. You'll notice these two very large tips right here. These are atmospheric water absorption features. And there's a number of non-pigment-based biochemical absorption features in this range, the short-wave infrared. My group spends a lot of time in that range. There's a lot of issues and logistical challenges when it comes to using those data, but that's where we spend a lot of time. The nice thing about this form of data is that it's scalable. Okay, so if you look, we have measurements at a leaf scale, canopy scale, and landscape scale from a manned aircraft imaging system, right? And while there are differences across the different scales, the shape generally looks the same, right? So you see kind of this characteristic dip where green is, kind of rises up, and then it starts to trend down in the shortwave infrared, and it's similar across all different forms of measurement. Theoretically, meaning that you can then scale the information from one scale either up or down to another. And what this allows us to do is to ask a number of different questions. So if we can calibrate spectral data from the leaf to the aircraft, or even downward from aircraft or UAVs to leaves, we can collect data across different spatial scales. So we can scale what we measure from a leaf at a very fine scale with control measurement up to a UAV or potentially even satellite information. And it kind of bridges this gap that we have from leaves to fields to landscapes in terms of questions we can ask. It led to a couple of different outcomes in the past 10 years, kind of theoretical outcomes, led to this optical type, where you can assess plant genetic and functional, you can characterize plants' genetics and functional variation using spectral data. And it's this concept of an optical type or a spectral fingerprint. There's enough biochemical variation and structural variation that's linked to genetics in plants that spectral data can collect, that you can start to build trees based on differences in structure and different compounds. So if this is chlorophyll, here's like a 3-flavonol subunit. All of those have different absorption features. Again, you can start to group plants into these different constructs. So how do we use this information? How does my lab use this information? We do a number of different things. It helps us understand plant-insect interactions. By training, I'm a plant chemical and physiological ecologist. I'll give you the reason I actually started using spectral data here in a second. It helps us understand environmental change, helps us kind of track biodiversity. And then we also do a lot of work in my lab in row crop agriculture, but we're not going to talk about that. So how do we use this to understand these data? And the reason I got into using spectral data, honestly, is because of pipetting. So I would go out in the field, collect 10,000 samples, run 10 different assays, 10 different chemical compounds, and have to pipette all winter. Nothing else was done. And it was killing me. And I said, there's got to be a better way. So I was working with a postdoc, familiar with the approach. We found the instrumentation and then moved forward from there. So it's kind of been, you know, it's trying to be more efficient and less, I guess, more lazy. But so there's 4 different ways that we use these data, and a lot of people use these data. One are vegetation indices. The second one I'll talk about are functional trait estimates. The third one, we have the spectral phenotyping approach, which is kind of closer to the spectral fingerprint. And then we can scale a lot of this information, make maps. Vegetation indices, I don't particularly care for them that much. I don't use them a lot. In many cases, they're non-specific and they can be fooled pretty easily. But here are some examples: green biomass, the normalized differential vegetation index. You can get an estimate of chlorophyll and nitrogen. Light use efficiency, so you can get plant physiological, water status. All of these have been developed. They've been developed for over 50 years. People have been developing these types of measurements. NDVI is a commonly used one to try and understand plant health, but I'll give you an example. And this is wheat, okay? And this was a very specifically chosen example because of what changes. So this is hecking fly, it's pest wheat. One of the ways to avoid damage is really through cultural practices of planting it at no fly time for the fly. There's a lot of efforts in breeding, right? And it's a very classical R gene mediated resistance, right? If you have the gene, the R gene, then you are not susceptible. What the fly actually does, and what we did is we infested plants, 2 sets of plants, some were infested, some were not infested, collected spectral data, We actually measured a number of other metabolites, but it's easy enough to calculate something like NDVI. And what we found is that in the non-infested plants, right, here's no flies, but these are susceptible genotypes. What we see is that NDVI declines. So you get this kind of developmental change, right, in NDVI. In the infested plants, that goes away. So NDVI stays the same. And what it actually does is the pest modulates host chemistry. So it increases protein content and decreases phenolic compounds to make it a more suitable host. So using NDVI to say this plant is unhealthy in this particular example wouldn't give you the answer that you're hoping it would. So functional trait estimates. What is a plant made up of? You have Nabisco, chlorophyll, a lot of structure, phenolic compounds, lignins, a number of different things. And how can we use spectral data to understand this biochemistry, right? Or these chemical properties and structural properties, and even physiology? Well, there's a number of known chemically important absorption features. So if you look at it, there's a number of absorption features throughout the spectrum as related to chlorophyll, pigments, proteins, phenolic compounds, leaf thickness, electron transport for JMAX. So a number, we can exploit those absorption features and relate them to variation in the spectral data. So what we'll do is we will do the standard reference measurement. This is, if you're familiar with milkweed, here's the toxin that is in milkweed. So we'll run the chemistry to determine levels of that particular compound, and we relate variation levels of whatever trait we're interested in with variation in spectral data using multivariate statistics. Partial least squares regression is probably, it's one of our preferred approaches. And then we can get a predictive model, right? So we can estimate this trace based on spectral data alone. So it allows us to go up to naive plants that we don't know anything about, measure them, and we can estimate these chemical properties. To date, my group, we try and run the gamut from primary to secondary metabolites to physiological parameters, anatomy, anything that you think could respond to stress. And the idea is if we can measure enough of these traits, we can get all of them with one measurement instead of the copious amount of time it's going to take to measure all of these compounds. Spectral phenotyping is what drives spectral variation in plants. We know chemistry does, we know genetics does, but we can't measure all the chemistries, right? We can't measure every single compound in a plant. We don't know all of the genotypes. We can't realistically know all of the different genotypes of all the plants. So if we believe in this spectral fingerprint approach, we can use the variation in the spectral data as a surrogate for this total expression, biophysical, biochemical, structural, physiological status of the plant at the time of measurement. So that spectral data can be used as an indicator of genetic variation for plant responses to stress. So we've seen that we have papers where we show we can delineate different genotypes of different plants, and we can relate the responses to stress. So we can use the spectral data as the vector of information of itself and kind of plug it into these classification and machine learning models to get the answer that way. So I like this particular example of this. When I was at Wisconsin, we were asked to go and measure population level responses. This is in Central America of live oak to different levels of water treatment. And a lot of the standard measurements were not showing any type of population of provenance level responses, right? So they invited us to come down, make a number of measurements, and again, our hypothesis, just as you would think with regular standard measurements, would be that the local environment of provenance would influence plant responses to water stress. And I'm cherry picking data from these two sites because they have the most disparate watering conditions or environmental conditions. But if you look, the plants from Mexico, and these are 4 different water treatments. So all the open circles are plants from Mexico. All the closed circles are plants from Costa Rica under different water treatments. It doesn't matter which one it is for the purposes of this example. But you can see that the plants from the Mexican province had much more spectral variation in response to the water stress. But they also had, a better growing environment, less severe stress, right? Where the Costa Rican plants, actually the spectral variation collapsed, suggesting that their response to water stress is probably more controlled and less variable than the plants from the Mexican province. So it's kind of this idea that we can see separation in spectral data that we can't if we say, you know what, I'm good at running a light core and I'm going to go and make a whole bunch of gas exchange measurements and that's got to be running this. Okay, there's just much more information in spectral data. So if we scale up, we can start to make these measurements at different scales, UAV based. We have manned aircraft based satellite imagery as well, and over the past 15 years, the introduction of more and more small satellites into the atmosphere allows kind of daily return times and 1 meter pixel resolutions. Spectral resolution is not as fine as you'll see here, but there are trade-offs for both of those, but we can scale this information up, right? So here's an example where we looked at, this is all aspen stands in northern Wisconsin. And these two particular compounds are from the same shikimic acid pathway, so they're both implicated in resistance or tolerance to insect herbivory. Okay, so salicylates, think of aspirin. If you eat too much aspirin, you get an upset stomach. Condensed tannins are just like a polymer of 3 flavanol subunits stacked on top of each other. They do a bunch of different things. But we went around, made a bunch of measurements, and scaled this up to satellite imagery. What we're able to do is map these compounds across these flight lines. And if you see this one, I mean, this is a pretty reasonable range of salicinoids in this particular area. So we can start to extend these to landscape scale measurements and then fold in other data to try and understand ecological processes. So we've done this a ton in row crop ag, right? That's kind of corn and soybean up there. We worked a lot in potato when I was at Wisconsin. We moved into cannabis, both for chemical detection and disease detection, and then a lot of work in maize and oil. But what I'm going to talk about today is more forestry. Okay? And the difference, there's a lot of differences between trees and corn. One being they're very long lived. 2, it's a lot harder to get to see over the top of trees. And so you have this kind of horizontal, which is this long-lived species, right? Combined with this vertical size scale of an issue. Because I can get on a ladder and look down at corn. I can't get on a ladder and look down at the top of a tree. So if we think about it, remote sensing approaches can monitor these systems. They can resolve some of these logistical challenges that we face, and we can start to work. I mean, I feel since remote sensing has been used in forestry for a very long time. In a management or breeding context, not as much. So if we can start to integrate some of the practices that are currently being implemented in precision agriculture or traditional row crop ag, they can start to move into forest management. There's a lot of questions that still remain, just as in row crop ag, about what information we can use to kind of blend into this precision forest management approach. So the rest of the talk is going to be going through examples from my students. So we have a couple of kind of larger goals in the lab when it comes to using spectral data in a forest system. One is to see what information we can get from tree biochemical, tree chemical physiological status, right? So we can start to build decision trees about how trees are responding to stress. And the second one is how do we scale these to different, to the tree stand landscape scale measurements. And I say we, but I really don't do a lot of stuff. These are a lot of my former grad students, current grad students, who do all of this work. We work at different spatial scales, so we're just as comfortable working in a greenhouse where we can control stress that we use very finely, to working outside in the field as well. The first part was really pushed forward by a former postdoc of mine, Dr. Lorenzo Petrozzi, who's now at the University of Pisa in Italy. And a current grad student, Sylvia Park, who's hoping to graduate next December. And to do this, we basically do two sets of work. We have our standard reference measurements that we would make. We collect tissue for chemical assays, we collect physiological parameters, we make structural and anatomical parameters. And then, as I was talking about earlier, when we blend those with the spectral data using multivariate models, we then can estimate those traits, right? Our goal is to see what information we can pull out. Nitrogen is an easy one. Leaf mass per area or specific leaf area, another easy one. Those seem to always work. It really doesn't matter what you do to the spectral data. We're interested in a lot of other responses. So physiological responses that are an emergent outcome of a number of underlying chemical parameters, or specific chemical parameters that haven't been estimated yet. I do a lot of work with secondary metabolites, so toxins, or things that would have an implication into a response to stress. So we collect, we impose stress on plants, we do all the battery of analytical work, all the physiological, water, chemical parameters, and then we make different various levels of spectral measurements on these. We do it at this scale for a very specific reason, because everything is controlled. We control the light angle, we control the measurement chamber, we control the light intensity. And our thought is, if it doesn't work here, when it's controlled as best as it can, why would it work outside when you introduce all the other variation? So this was driven again by Lorenzo and Sylvia, and I'm gonna quickly skip through a lot of these. We went to a whole bunch of experiments and stressed them out in a bunch of different ways. There was water stress, salt stress. We didn't fertilize some. We put a whole bunch of salt in the soil in another one. We measured all these traits, and the goal was to see what could we model using the spectral data, right? And so I'm going to click through these. Don't worry about the actual parameters on here. What I want you to look at is from the bottom side to the top right, bottom left to top right for each of these, right? So these are gas exchange and chlorophyll fluorescence. So you have things like photosynthesis, somatic conductance, water use efficiency, leaf temperature, and chlorophyll fluorescence. Look at the bottom left, top right, right? Most of these traits we were able to model pretty well. We were pretty happy with these outcomes, okay? So we look at water status. We have things like water potential, osmotic potential at full turgor. All of these are important implications for how plants respond to drought stress, water stress, or flooding, relative water content, leaf thickness, right? Again, if you look at the bottom left to the top right, most of these functional traits were actually well predicted by spectral data. And we get into some primary and secondary metabolites. And this is just a snippet. I think Sylvia is up to now 56 different, just chemistry alone combinations. She has about 22 individual phenolic acid and flavonoids that she's trying to model. But you can look at things like nitrogen, chlorophyll, individual sugars. You have flavonoid rutin over here, gallic acid, phenolic acid. So these are examples. And again, most of these are well predicted using spectral data, right? And the idea being, what if a stress actually presents itself in different ways, right? So what if you have a stress that co-occurs with another stress, but you see the same response, right? So say you have something feeding on the roots of something, and you notice it starts to wilt, or you have a wilt pathogen in your tree. How do you know it's wilt, or something feeding on the roots, or a root pathogen, or something, or water stress, right? The idea is there are subtle changes, whether it's through chemistry and physiology, that plants will respond in a coordinated way, okay? And if we can start to layer all of these and to say, yes, the spectral data indicate that these plants are healthy, and because and use a decision tree algorithm to say these 7 parameters changed, it should be the probability of it being resilient is high, right, for oak wood. So that's the idea behind trying to measure all of these different traits. Once we get these traits, we can also use just standard statistical approaches to understand how these plants are responding to stress. And here's an example. I mean, we plug them in to just standard, linear models, and we can get similar results if we focus on this particular one, water potential. We see that if we have our observed trait, this is all that was measured. So these are all the measurements we made. So this is a water stress plant. This is one that we get very happy. We see a similar response from our spectral trait estimates after holding out the observed values, right? So what we're suggesting is that you can use these as a surrogate for the actual reference measurements in any type of statistical approach, right? So instead of 800, right, pressure bomb measurements, you can do 100 at different time points, build a model, and estimate all of the ones that you've collected spectrally. If we go into this idea of spectral phenotyping, right, if we move into do spectral data separate, is there variation in the spectral data? We can use that too. So here's two just really brief examples of a drought treatment and a nutrient treatment, and there is a lot of overlap, but you can see that the spectral data do separate. So imagine this being the first layer in a decision tree, right? It looks like something's wrong. It could be drought, it could be X, Y, or Z. Then you layer on all the other functional trait responses to try and get at a higher probability of coming up with the correct stressor that's correct. So we're able to kind of classify these things and see how they respond to different treatments over time. Okay, I'm going to move away from kind of leaf level measurements right now, move into UAV-based measurements. This was primarily done by two postdocs that I had, Dr. Alamasjetti, who is now at Bayer Crop Science, and Dr. Baruch Naziri, who's now at NASA. And where I live is like right here, let's say, right? There's a plantation. There's a huge push for regenerative chestnut growth, right? If we can get a resistant chestnut from chestnut blight, there's a lot of groups that would like to see it planted. Historically, American chestnut was a foundational tree species that was very widespread in the East. It still exists on the landscape. I still see it. It just looks really sad because blight kills it, and then you get stem dieback, and then you kind of get this suckering until it gets big enough and blight eventually falls, right? And if you remember where we were, here is one of our land holdings for FNR at Purdue. Here is our forestry center off campus. And up in this corner, what we have is in 2005, there was a mixed species planting. And really the goal was to see, okay, if we plant 3 either economically or ecologically important tree species, do they grow better together, right? I mean, some trees act as trainer trees to grow more expensive trees straight. That was the idea. So this was oak, chestnut, and black cherry. And what we were interested in about 2017, we saw chestnut blight starting to kind of come in, right? I hadn't found it until it was probably about 10 years old, and then it moved in. And these particular spots are all the different individual chestnut trees that we identified and that we used in this study. So we took this opportunity to say, I wonder if we can track the spread, the development, the epidemiology of chestnut blight as it's moving through the system. Can't do anything to stop it. So we might as well measure. And what we did is went out and we scored chestnut blight. So we had a numerical 4-score system where we classified what level of severity was. And on the right, if you look at it, this is the number of trees in each of these classes, right, through time. If you look in 2018, we had very few trees that had blight, and you can kind of see this shift, right, to in 2020, there were very few trees without blight. So it was moving through this system. Along that time frame, we also collected UAV full range hyperspectral data, right, at 3 times a year in 18, 19, and 20. To do this, we use LiDAR to create a vertical or horizontal surface of individual trees. And then we extracted those trees and the pixels from those trees from the UAV imagery to compare them across years, across time points. We normalized the data, so basically that's just setting it to 1 is the most, and then everything else is less than that. And then we kind of, it makes it a little easier to translate or to compare June to August, and especially as we move across years. We then ran it through a pretty basic support vector machine. The whole spectral data went into this support vector machine classification algorithm. We also subsetted bands for our input variables to see if band selection had any approach. We used it based on the correlation cutoff that we had for each band with severity. And then we just set the threshold higher and higher and higher to reduce the number of bands, the input variables, to see if there was any effect. Didn't look like there really was on which bands we used. But the best bands were in every model, right? So it's kind of, you know, it's cooked in there. What we found is pretty good classification across dates, right? So for all the years, 18, 19, and 20, we get about a 70-ish, 74.5% classification accuracy. And you can see the confusion matrix here, and this is if we just have 2 classes. So remember I said we scored it in a 4, this is just a 1 or a 0, right? This is either you have it or you don't, okay? And we see that it worked fairly well. We then moved to 3 classes, and it drops down to about a 62% classification grade. And you start to get some confusion in the middle as it comes along on whether it's a 1 or a 0 on some of the edges. But again, band selection didn't seem to have an effect on that as well. We then put all 4 in, all 4 classes. We see it drops pretty considerably to just above 50%, right? Bands didn't really have much of an influence again. And you can see that the confusion matrix, the numbers are becoming more even within the matrix itself, right? And I like to think of this as the sport effect, okay? Everybody knows what a spoon is. Everybody knows what a fork is, right? A spork is both. And if you have something that's in between a 1 and a 0, like 0.5, it's going to be harder to classify than if you just have a spoon and a fork, right? Think of it from just a visual standpoint, you can definitely delineate the spoon and fork. But once you throw in something that looks like that, any type of, you know, machine learning algorithm is going to say, it looks like both of them. I don't know what it is, right? I think that it's like the loaf of bread, the dog, the kids movie. It's a good one. I'll forget it. It's this pug that Mitchell's saved the universe, I think. And
Mitchell's machine.
Mitchell, yes, machine. And the dog is classified as either a loaf of bread or a dog. They can't tell which it is because it's this round pug, right? It's like big and brown. So that's another. Now I'm going to click through these. The most important thing, we watched this through time, and this was really the tell, is that could we track the shift in development? If we look at this, just look at the color change, with lighter colors being less severe, red colors being more severe. And if we relate it back to that distribution of scores throughout time, we can see that we had very little blight across times in 2018, across years or across months within each year. We start to see that there's development, more trees have light, and then we see that light is pretty predominant in most of these stands. And it doesn't matter which score we're using. What we can do is, if we track this to actually relate this to our scores that we measured, we see significant positive relationship, meaning that our reference scores are being supported, are being supported by the classification. All right, I'm going to really quickly switch. I want to really quickly switch to a new student that I have. She worked with me for her master's, is now a PhD student in FNR. And this is using spectral data to detect spot and lanternflies, right? Lanternfly, not here yet. Will be. Not here yet. Introduced in Pennsylvania, 2014 on a stone shipment, and it's spread west. We found it in 2021. That was the first. It was along the shipping channel in southern Indiana. We've since found another population in northern Indiana along the railway. That's a common theme of how they move. But if you look at temperature modeling, this is the kind of projected distribution of lanternfly, and we're right here. So it could exist in quite a few spaces with us, but there's a good amount in the eastern United States that could support lanternfly establishment. It's a phloem feeding plant hopper insects. It's very colorful. It's also very quick if you ever see it. Like it jumps really quickly if you're trying to catch it. So one of the questions he has, does lanternfly influence canopy spectral properties? And we did this in collaboration with Penn State and Temple. So we're funded to kind of try and map species, lanternfly distribution and develop probability maps of where they actually are more likely to establish. But the first thing we had to understand, is there an individual tree response? If we know that lanternfly is on something, we can control the numbers. Do we see a difference in spectral data? And again, there's a common garden out at Penn State. We did this by caging individual trees. This particular study was done on maple. And then we did a multi-species enclosure that had 4 different combinations of tree species. Now I'm just going to pull out point data from each of those, not the entire, because Ellie's still putting together the story. But if we look at the single species enclosures, here's the spectral profile of each one measured, and you can see there's definitely separation in multiple spots between the treatments. Normalized differential spectral index is a pairwise combination of all spectral measure, like all spectral wavelengths, right? So it's 400 minus 2400 plus 400, or divided by 400 plus 2400, right? So it just generates a differential index. But what it can do is identify areas that are more related, then relates it to an input variable such as density, and you're able to get areas of spectral of the spectral profiles that are more effective, right? The closer it is to this one-to-one line right here, the more it's actually that wavelength. When you get out here, it's actually interactions with kind of shorter and longer wavelengths, which is a little more challenging to interpret. We see a lot of shifts. If we look at relative shifts on the lower left, we see a lot of change here in this visible wavelength range right here, right? 800. Just after actually the visible wavelength range in what's called the red edge, which has historically been used for quite a bit as an indicator of plant health. Then we also see shifts in different wavelengths. And we kind of cut these out because in our separation model, in our clustering analysis, we found these produced the best result. And these longer wavelengths we actually found are related. There's a very characteristic absorption feature for phenolics in here, and one out at about 2200. And so, because we did the chemistry on a lot of these plants, and I'm pulling out a few, but what we see is this particular phenolic acid, ferulic acid, right here, increased at low and medium densities, and then dropped. Gallic acid, which is a common response to oxidative stress for pathogens, also increased. Ferulic acid is a very characteristic plant response to pathogens, right? And in many cases, phloem feeding insects upregulate the salicylic acid pathway for defense, which more mimics a pathogen than the jasmonic acid pathway that's upregulated by most other herbivores. There were a couple other shifts. We saw some shifts in flavonoids. This was an interesting one where we saw declines or decreases in tranexamic acid, which is actually a precursor to ferulic acid. So we're starting to kind of potentially put pieces together that indicate that this precursor, because these exist in pools, this precursor was converted into this and differentially so at different lanternfly densities. So that's a really interesting outcome that we thought. So we have these multi-species enclosures. I'll walk you guys through this. On the left, we have the average lanternfly count. So in these enclosures on the upper right, there are 4 trees. I just plucked 2 of them out that I want to talk about. Every tree is measured monthly, so we get 4 different measurements, right? These particular lanternfly densities on each tree were counted at the same time. So we get to see their movement from tree to tree across the season. There's thought to be some host switching throughout the season based on host chemistry. And what we see if you look, lanternfly numbers peaked in July, right? And then they started to decline. And what we see is if we look at this circle is this circle, right? After we see the largest infestation of lanternfly, and then they decline, we see a spectral separation that persists throughout the growing season. There's potentially some legacy effect. We're still running the chemistry to see if any of the former stuff actually holds up. But there seems to be a legacy effect where, and this is tree ahead, so this is Atlantis, one of its preferred hosts. There seems to be a legacy effect that we're picking up using spectral data. Another preferred host, black walnut, which is one of the trees that we work quite a bit on because it's economically important. After we see a spike in lantern numbers, right, you see that this separates out from the other ones where lanternfly numbers decline, and these become more similar to each other. So this appears to be a more immediate response and then not responding afterwards. We don't know what that means, but we're hoping that the chemistry kind of give us an idea of what's changing. Next steps for this, like I said, we're working with Penn State and Temple to kind of update a lot of their probability maps to include kind of stand level spectral characteristics from small set data, right? With high return time, good pixel resolution, usually about a meter. It's multispectral data, but if we can get an idea, some index of habitat quality, and overlay that with the current environmental like MaxNet modeling that they're doing, then I think that's got to help improve the probability of where we think something may be able to establish. So there's a lot of different stuff we can do with spectral data. You know, we can rapidly and non-destructively measure chemistry, physiology of a number of trees, and a number of plants in different ecosystems that are otherwise difficult or logistically challenging to obtain. We can make measurements in vivo, right? You don't have to go and take a branch off something. We can monitor plant responses to stress. We can increase data volume collections. It's a lot easier for me to walk around or students to walk around and make measurements than it is to clip and go through the process of correctly storing the tissue, spatial variation of plant functional traits, and how that relates to ecological processes is an area that we're interested in. We can map those traits, and we're actually moving into a lot of breeding efforts with these data. So understanding plant responses, specifically with walnut, responses to water stress, how does that in fact affect like climate resilient tree breeding? So we're trying to get that. With that, I'd like to thank my lab, thank Dave for inviting me. A lot of collaborators and a lot of different funding sources. We have a great Center for Plant Biology at Purdue. We now have an Institute for Digital Forestry. We're part of the CAF Center for Advanced Forest Systems through NSF. Our Hardwood Tree Improvement and Regeneration Center funds a lot of this work. And with that, I'd be happy to take any questions. That's my oldest right there on our list. That is, he's a lot older than that.
I was gonna say that's an old,
Yeah.
an old picture.
Yes, he's 10 years.
Real quick for the forestry folks, you mentioned trainer trees. Can you explain trainer trees when they're used? If we don't,
Oh, yeah, sorry. Different parts of the world. So the thing with trainer tree is something that's going to grow faster than your economically valuable tree and train it to grow straight, right? So if a metric of tree value, right, is forward length and straightness, then if you can grow a straighter tree by having trainer trees around it, that's going to be much better than letting it grow by itself. It's not going to compete as much with those trainer trees, and the idea is just to get a better economic return
Something like, when you guys are growing something like walnut or cherry for veneer.
Yeah.
Right, and you want a perfect straight branchless tree, you put, I don't know, what species would you use trainers?
They use a lot of pine.
Pine ash, so this pine's just gonna shoot it up, and basically just. Do guardrails around it.
That's pretty much what it is.
Questions?
First, thanks, that was a great presentation, and as a non-chemist and non-botanist, non-forester, it's blowing my mind, all these things. I was curious about, when you showed the outputs from Mexico and Costa Rica, and you kind of had, same plants, different environment. I'm really curious about the variation that you're seeing. And do you think there's a pretty broad range of intra-species, individual variation and responses to things that you're gonna see in the spectral?
Oh, yeah, I would argue that there's as much intraspecific variation as there is intraspecific.
Yeah.
So I've seen as much variation in, F2 hybrid vine corn as I have in disparate trees like oaks and pine trees in the spectrum. So it's considerable.
So is that like you wanna essentially test enough that you can find common patterns across things when you're looking at signatures that you could make some?
So that particular study, we were interested in whether or not your product, your point of origin,
Mhm.
your historical climate conditioning influence was affected your response to different watering treatments.
Mhm.
So that it's not quite, it's a hard comparison to make what you're asking because they had a pre-treatment, right?
Yeah.
They grew, or the seeds were collected from a specific area that had, the parents had very specific environmental upbringing, if you will, right? That's why I like to stick with the chemistry, because that makes a lot more sense to me. But we do it both ways. If you see a difference, right? Think of a tiered framework of responses that you can have. Right? If you identify an area of a field or a forest or anything that looks different spectrally than the other fields, one outcome may be we need to go and go and take a look at that little pocket, what's there. You can then go and assess whether visually, depending upon the goals of the individual making the measurements, you can then go and make finer scale resolution measurements, right? And then use that information to come to the decision.
Thanks.
What else? The Zoom question there.
The quick question. It's sort of simple and not really that, but first I thought it was a very digestible. I'm kind of an errant but where like I'm not exposed to any of this and I was following it and I really appreciated the way that you condensed that info. So it seems like there's a lot of agricultural applications, and so who generally tends to be like the recipient of this info, because is it sort of like overarching agencies? Is it people who would have large plantations? Like who kind of would use this in like a whole setting?
I mean, most of forest holdings, at least close to where I live, are private. So it would be private. There is a technology gap, right, of getting through. This may be more information. Right? That doesn't mean that Bear is interested in that information, or that Hauser or Pike or somebody is because I've had those conversations with them as well. How do you use this? I mean, I'll give you an example. There's a software package, I can't remember the name of it, that comes with the UAV platform that goes and calculates indexes in real time. You can watch it on the tablet as it's flying through the field. Right? It's done over row crops. But imagine the same thing going through the forested area, and if you can calculate some index of health, then that gives you in real time. You can automate those things to fly twice a week. Then you just get that information sent to you. So the level of information is dependent on the end user and what they really want out of it, and whether it's cost effective as well. But I think That's the goal, at least for us, some kind of, part of a sustainable agricultural systems grant that's trying to transform front to back end of forestry. One of the approaches is technology driven, where it's tree identifier from drive or health identifier from like a lot of really simple tool-based approaches that can be transferred easier than here's an entire software package that needs these different layers, that needs these different inputs to get to the answer.
I have a really quick question. I know almost nothing about plant chemicals. But has there been any crossover application of this to like mass production prediction? Like in any of the spectral data where you might be able to directly apply that to, you know,
Yeah.
what's a predictable crop of acorns for
Yeah.
species? Because that could then be like related to bird wildlife
Mhm.
prediction.
Oh, absolutely.
So,
Yeah. Yeah, I don't like to say no to any question, right? Especially if I don't know the answer. But it seems like if there is a physiological response to the tree as it's prepping to nest, and you can capture that over enough. The thing with trees is they live forever, right? Well, not forever, but they live a long time, and you can actually collect those data on trees. You have different nest events, you know, at different times of the year. So I don't see why you couldn't. What I don't know is the physiological responses that actually occur when it leads up to nest. So assessing that would kind of be, and is that physiological response, is the magnitude of that response strong enough to detect something?
So just, I kind of think of that in the framework of, you know, replanting for chestnut, if that ever gets to the mass scale of original coverage. You know, there are a lot of species that then people think, oh, they were driven historically by the chestnut bumper crops, if you will. Or if there's any like application. So,
Yeah.
Thanks.
Yeah, I'm from the field forestry and I'm beginner engineer too, so remote sensing is also something potential. So my question is pretty basic. I don't know the difference between like remote sensing and like how like is a hyperspectral remote sensing can output from the regular remote sensing. And like I was thinking like, is there a way like that we can kind of get information about the regeneration of trees which are lower layer in forest and with a hyperspectral, can it go deeper?
So penetration through the canopy is always a challenge when you're making a broad canopy. So that is one of the limitations. You're usually focusing on the top. That doesn't mean that you can't time the acquisition of events such that overstory canopy is not flushed out, the understory canopy has. Right? So if you're trying to track understory species, leaf flush and movement, then do it really early in the season before the canopy, the overstory canopy is flushed out. It's still going to be a bit messy, just because you'll have that structure there. But if you overlayer it with something like LiDAR, you can mask out the actual structure of the tree and then segment out individual pixels, then you may be able to get that. Additionally, we fly them under the trees as well. You have an issue, especially depending on the time of year, the light intensity that you're getting. This is all passive remote sensing. So this all depends on the amount of light that actually gets down to the bottom of the canopy. So that is one issue. But I know a group at Purdue that's working on making those measurements underneath the canopy. So it's more for production forestry, so things in lines may not work as well in natural systems. But I think if you kind of pick and choose when you make your acquisition, it may be possible, be more possible than the middle of the summer.
So y'all had success in following around the moth. Do y'all see any interest in things like the US government trying to do stuff for Asian longhorn beetle or southern pine beetle?
Yeah.
We can get data like once a week pretty good.
Mm-hmm.
So like you could theoretically just automate that and just have it spit out and show you
Yeah,
data.
Dave and I have had this conversation a couple of times with, fine systems or ALB. ALB is a little trickier in my mind. Just because it has different hosts, right? But there's no reason you can't. And with the advent, again, of things like small satellite data, which have daily return times, and very fine scale pixel resolution, I don't see any reason, I'm not a computer scientist, so I think all of those things should go together. How they actually are put together, is a different story. But yeah, I don't see any reason why you couldn't automate imagery to be downloaded and then moved into, I know you can get real-time planet data from as a company that has small set data and feed it into whatever other input variables you're trying to load into a model to then try and predict probability.
You kind of touched on this briefly, but is there a way you can control for different like lighting conditions? If it's like a cloudy day versus for a sunny day or like different times of the year, is that going to affect?
Yeah.
Okay. Is there like a certain like? control
There's a
you
number
have?
in the remote sensing world, there's a number of people who work on how do you get around that issue, right? And I don't know a ton about it. I know it's a big problem, right? Some images are not able to be used. It's fine. Right? You just have, you can't, or you can mask out the clouds and just use what you can see. But it is a problem, right? But there's radiometric calibrations that are employed to try and say, look, it should be here. Let's bump this up to this. For a lot of the controlled studies, you put a very reflective white panel out in the corner of a field. And when something flies over, it captures that in the image. And we know what the reflectance from that panel should be, which means we can back adjust everything to that panel. Not the easiest thing to do, right? In the middle of the Amazon if you're trying to monitor things. But there are radiometric calibrations.
What else? So, John, thanks again, man. Appreciate it. Thank you. There will be a feast for graduate students. Use that term as you will, but I think it's in this room, right?
Yeah.
All right. Would you or you get him in my office when you're done? Please, thanks. Or you, what am I? Hold on. Just a little laughing. It is.
Yeah.
I know. Everybody's like crashed, right? So